the whole team at the HPC. So my name is Vereecke. I am actually a PhD student at Ghent, but I also work together with Pathosense, a new diagnostic spin-off of the UGent at the veterinary medicine faculty. So I will talk in general a little bit on what we are doing, and I will start with a little introduction on what nanopore sequencing really is and its applications. Then I will, in short, go over some SARS-CoV-2 because we all are aware of the corona things at the moment. So we did quite some work on this as well. So I want to share this and how we used HPC with that. And then my major part of my PhD is actually bacterial whole genome sequencing. And there I will also show how we use actually the HPC in generating genomes and what the importance of the supercomputer is here for us. So first of all, what is actually nanopore sequencing? I will actually make the comparison with Illumina because many people will know what Illumina is. Illumina is rather a short read and a golden standard sequencing approach. And I will actually go over this table in little steps to make it clear what the differences are between actually the different techniques. I will not go too much into detail in Illumina, but I will show the actually interesting part of the nanopore sequencing. So first, how does it actually work? For that, I'm actually going to show a little movie here. You can see it's a very small device. It's actually a kind of device that you can compare like a printer. It has a little flow cell inside, which is like the cartridge. And there we will load the DNA. Here you can see the blue is the DNA or RNA, and then we will add some proteins to that. And we are simply going to load this actually into this cartridge. Inside this cartridge, you can see the little circles there. These are very small pores. That's also where the nanopore sequencing comes from. These are actually going to be very important for this case, as the DNA is actually going to bind next to these pores. So there you can see how this is going to work. What is next going to happen? The DNA is actually going to flow through this pore, and as we compare with Illumina, there we actually have the synthesis with the enzyme that's going to happen. Here we're actually looking at the signal by the voltage change that there's going to be by the DNA going through the pore. And this will give a signal, as you've seen on the picture, on the movie. So this will continuously happen. And actually what is then going to happen at the end, we get a raw signal, and we can easily convert this, as you can see here, to the nucleotides, which is the genetic code A, C, T, or G. And this can actually at the end give us the interesting data, the same as we would have with Illumina, but there's other advantages as compared to actually the Illumina sequencing. So this is how the technique actually works that we are using. Now, one of the biggest differences is that nanopore is long read sequencing as compared to short read sequencing with Illumina. As you saw, we work with the pores, and there we can actually have very long stretches of DNA flowing through these pores. Illumina is really fixed because we use an enzyme, and there this enzyme will run in different cycles, which has a specific time interval, and this will only generate up to 300 base pairs of data at one cycle. How can we compare this? If you look here on the left, we have the long read approaches. On the right, we have the short read approaches. If you would have to look for Wally, for example, as you see here, with the long read approaches, we would actually need less data to actually get the entire puzzle fixed. If we need the Illumina, the short read approaches, we actually will need way more data in order to have every part of the puzzle covered. So that's the biggest difference, and a quite interesting one if we want to go into genomes like bacterial, viral, but also human and other species genomes. Now, the read quality has been always the big problem, and everybody that is a Illumina believer or a nanopore believer, this is the everlasting struggle. However, I know that this has changed over the last years really in a very good way. And this year they actually promised that we will reach above Q20. Now, what does a Q20 actually mean? Q20 means that there's 99% accuracy. So this actually means if you have 400 base pairs, at the moment it's 98.3, which means if you have 400 base pairs, you only have 7 mistakes in there. If we go higher, 5,000 base pairs, this is 85 mistakes. Now, if we compare this to Illumina, we have 99.9% accuracy. So that's actually 10 times higher accuracy than 100 times better accuracy. However, we're talking here about the single reads at the base. At the end, we can improve this still by actually sequencing deeper and not looking at only one single piece, but we will actually sequence one single part of the genomes multiple times to actually get to a better and higher accurate genome. One other big difference is actually the instrument cost between the two techniques. We have Nanopore, it's actually quite low in cost as compared to the high upfront cost you need for the Illumina sequencing. And there's different ways of actually buying, there's different instruments available. We have this giant Promethean over here, which is also rather in the more expensive way. This can generate a lot of data. We have the GridION, which we will have soon here at our lab as well. This is actually a smaller version, but it can also already generate 250 gigabase pairs data actually. And then we have here the smaller version. That's the one I showed you in the little movie. That's the portable one, which you can also use in the field anywhere you want. They actually sequenced on the moon already with this MinION. And then there's the very interesting one as well, the Flongle, which is a disposable sequencer actually. It can not generate that much data, but it's interesting for a diagnostics approach. Now, you see a lot of numbers just for you to give an idea what can be sequenced with which device. If we use this machine, we can actually sequence the human genome 2,000 times. If we use this machine, we can sequence the plant genome. I used here Arabidopsis. It's one of the model organisms, 1,500 times. If we sequence with the MinION, we can sequence 1,000 times the E. coli genome. If we use this smaller one here, we can actually sequence up to 90,000 SARS-CoV-2 genomes. So just to give you an idea of the different possibilities of the different machines. Now, one of the last things that are quite interesting with this technique is that the versatility is quite high as compared to Illumina because it can sequence many different things. It can sequence native DNA. It can also sequence PCR amplified. So if you have very low input DNA samples, you can also amplify this using a PCR amplification. But you can also start with RNA and then convert this actually to DNA, and you can sequence this. So far, nothing new. You can also do this actually with Illumina. However, there's some interesting things with Nanopore that we can also natively sequence RNA. So we can really go and look to the RNA that is in the samples. So you can go to RNA viruses, as for example, the coronavirus. You can really sequence the RNA from the virus without needing the conversion to cDNA. Another really interesting thing is that we can look at the modifications onto both DNA and RNA. This is one of the techniques that is interesting for that, as we don't need to manipulate our DNA or RNA at all. It's in the signals, as you saw. It goes through the pores, and if there's a little modification, we are also able to actually get this from the raw data. So within one experiment, within one sequencing approach, you actually can get a lot of more data than what we would have if we would be doing another sequencing approach. Now, you might be wondering, why do we need nanopore sequencing? As I said, I'm working for a diagnostics company, Pathosense. It's a spin-off of the UGent, where we actually at the moment have a complete platform available to use actually sequencing to perform diagnostics in veterinary medicine. We can use any host here, any sample time, and we actually don't need any selection in advance. If you go to the doctor and your doctor needs to do a test, let's say now the corona test, then they really have to look for the coronavirus. So they will only look for the coronavirus, but if you're sick from any other virus or any other bacterial species, they won't identify this because they really search for the coronavirus. Now, with this approach, we actually use a metagenomics approach. We can very quickly see anything that is present within one sample. Is it a virus? Is it a bacterium? Or is it a mycoplasma? Mycoplasmas are somewhere in between the viruses and the bacteria. Now, at the moment, we can perform this identification, and in the future, we actually want to link this also with antimicrobial susceptibility testing, which means if we have a bacterial infection, that we can also tell the veterinarian or the doctor in human medicine which antibiotic to use and which one definitely not, as there's many bacteria that have some resistances against specific antibiotic treatments. So for the introduction. Now I want to go a little bit deeper into COVID or SARS-CoV-2 work we've been doing. I will also give a little introduction to this for who didn't hear a lot about Corona yet, but I think everybody heard a lot about it. But this is going to be more about the molecular and the sequencing side, and then I will go into the use of HPC in the sequencing efforts. So the genome, it's about 30 kilobases long, and it's a positive single-stranded RNA virus. Actually, what was quite interesting is that immediately in December 2019, the first complete sequence was available. So we could immediately see what was inside this genome, if there were specific features, what were the resemblances with other viruses, and so on. And very shortly after that, there were standardized protocols available through the Arctic network. It's a network that has been active in Ebola sequencing and so on. And they immediately actually delivered a protocol with Josh Quick, James Ferguson, and Nick Lohman at the head of these developments to actually give all people all over the world a standardized protocol to quickly sequence in high throughput all the SARS-CoV-2 genomes. And that is the same that we did in Belgium. Now, again, here, the question is always, what about accuracy? I already told you that it gets higher and better over the years, but of course, we are not looking at single reads. I will explain a little bit more with this figure. If you have the genome in black here, we get single sequencing reads in blue. So it's one piece that actually goes through the pore. And you can see here that in green, we have a true mutation, but in red, we have a sequencing error. I told you if there's 400 base pairs, at the moment, there's the chance of having on average 7 mistakes in one read. Now, of course, we're not going to sequence only one read. We're going to sequence many reads. So as you can see here, if we actually pile up all these separately reads, we will be able to distinguish the true mutations, which will be present in, let's say, 99 to 100% of the reads, from the errors of sequencing. So in the end, we will be able to tell that the green mutation is a real mutation that was not present in the original strain that was sequenced in Wuhan in December 2019. So that's actually the idea of the sequencing. So we didn't do it for one piece, but we actually divided the whole genome into all separate pieces to amplify these very easily. As you can see here, we have the whole genome on the x-axis, and we divided this in approximately 90 to 100 sequencing amplicons. As you can see here, the read depth, we sequenced between 100 to 1,000 times each of these amplicons to get actually to this accuracy, to elevate the accuracy. And that was what was shown actually in the figure here, that we need, this was compared to Illumina sequencing, to get to a really similarity and the sensitivity of detecting mutations, we need to have at least a 50 times coverage. So 50 times the same region sequenced in order to get to the same level of accuracy as the the Illumina sequencing. So yeah, we, with our lab and together with the lab at the UZ from Bruno Verhasselt and Laurens Lambrechts at the HIV Cure Center, we started actually immediately in March 2020 with a small scale sequencing. There were only a few samples available, but we immediately said, let's do it, let's start with it. In Leuven, they were also busy with it. In Antwerp and in Liège, Luik, they were also busy with it. And here you can actually see our first sequencing run being available on the 27th of March. Since January 2021, there was a big upscaling of this because of the government. They wanted to put a lot of efforts in this, so they actually released a lot of money for sequencing labs. And then actually this entire SARS-CoV sequencing went to the UZ Ghent, where they have a big team on it now. And actually June 2021, so I checked this last week. At the moment we have almost 30,000 corona genomes that have been released from our country, so from Belgium. This is actually not a lot because this is only 2.59% of all positive cases in Belgium. So the sequencing rate is not that high because actually upscaling became actually quite late. Now, if we look at other countries and the sequencing efforts there, we actually have Belgium here somewhere in the middle, and the biggest efforts are done in Denmark, Germany, the UK, and the US. However, if we really look at the percentage of sequenced cases, Denmark and the United Kingdom are really heading up. In the United Kingdom, they sequence 10% of all of the cases. In Denmark, this is even 38% of all cases, which gives a lot of data, of course. And you can see here in red, 2 million complete SARS-CoV-2 genomes are at the moment available online because people can deposit them on GIS8. Now, why is HPC important in these sequencing efforts? Of course, I told you 2 million genomes. More genomes is more data, is of course more computational power required, because the sequencing output, it doesn't result immediately in a COVID genome, of course. If we make a small calculation here, if we have 30 kilobases per genome, 2 million genomes, we actually end up with about 61 gigabases, which is about 65 gigabases of data if we consider other additional lines in the data and so on. But this is really the final genomes we're talking about, because in order to get to these final genomes, we also need to construct a genome, because this is not the raw data we're talking about, which is still going to be much higher than this 61 gigabases. We want to do some phylogenetic analysis here, which requires bootstrapping to give some significant power. And then there's also the possibility to do some time-guided phylogenetic analysis, which requires GPU versions actually in order to fulfill this for really big data sets, as is the case here for the 2 million genomes. Now, the genome construction, why is it important? As I said, we have actually, for each genome, we have split this in 90 different pieces of each 400 base pairs. As I told you, we need at least 50 times coverage. So this means that 400 base pairs times 50 means for one genome, we actually get 2 million bases, which means 2 gigabytes of data already per genome. So this is not a final genome, but really the raw data that actually comes out of the data of the sequencing. Now, we don't perform one genome at once. We multiplex this to save costs. And we actually get 24 up to 96 genomes per sequencing run that can be produced within 24 hours. And this delivers actually about 48 to 200 gigabytes of data that is processed for only one run of 24 hours. Imagine what this would be if you count all of this together for this 2 million genomes that have been generated worldwide at the moment. Now, phylogenetic analysis is important for us because we want to compare the different viruses and we want to see how they evolve from each other. So we know the founder virus, which is the Wuhan strain that was circulating in 2019, December. But of course, you've all heard about the variants of concern and all the Delta variant we are talking about at the moment. And they can identify this by looking really into these genomes and identify specific mutations within one genome. Now, to do so, we need to compare actually every SARS-CoV sequence one by one. This is also called multiple sequence alignments. Now, if we have 10, 20, 30 genomes, this is not a big task to do. But of course, if we're talking about 2 million sequences, this is becoming a very, very hard task to do, which will require a lot of computational power. So we can actually cluster all the closest or similar genomes together to actually generate a phylogeny. And then at the end, we're going to add some significance by repeating this actually procedure 100 times, at least 100 times, to see if this clustering ends up always in the same way or not. We can also add the time of the sampling, which is then the time-guided phylogenetic analysis. Now, I will explain this a little bit with the figure below. We have a mutation rate of about 2 mutations per month for the SARS-CoV-2 virus. So if we have here virus A, which can be the Wuhan strain from 2019, we can here see that there's a mutation in red and there's another mutation in green. So these two viruses actually evolved in a different way. So there might have been an evolutionary pressure and there might be the spread of the virus. And let's say this virus went to the US and this virus came to Europe. And in Europe, this variant will actually spread further and the other one will spread in the US. So that's how we can compare the different viruses amongst each other. Now, of course, it's not about only these two. We will have multiple genomes at the same time. So if we have two genomes with the same mutation, they will end up together in the phylogenetic tree. But of course, there can also be already viruses that have a new mutation as compared to this first mutation here. So if there's an additional one, we know it has the green one, so it will be in the same cluster as the green, but it has evolved a little bit further as it has an additional mutation. As we know, there's approximately 2 mutations per month. Now, imagine again, this doing times 1,000, times 10,000, times 1 million genomes is going to take us a lot of time, and that's why we actually need to use the HPC for this. Finally, if we really want to add the dates to these trees, it's still going to add an additional complexity level to the analysis. So it's not only going to look at the multiple sequence alignments, but it's also going to take the date of sampling into account. So here you can see a real-time example from our samples in March and April 2020, where we actually had the first sequences of Belgium here in yellow. And the interesting thing about these kind of analysis is that we could see that there was actually a close link to the Netherlands at that moment. And we could actually see that the patients that arrived at the UZ in Ghent, that many of them right before the lockdown, they still quickly went to the Netherlands actually to buy some stuff and so on before everything closed down. And this is what we saw, that there was actually the same virus circulating in the Netherlands as was circulating in Belgium. So this is one of the things we can look at. Now we can look in a bigger picture here and we can do some outbreak analysis. You have some outbreaks you can see here in different colors. This was the pink one, was a very big one. How do we know it's an outbreak? Because there's no other strains present that we have sequenced. So this is really one cluster which was found at, this can be in the healthcare centers, at the hospitals and so on. And one of the biggest ones was in Belgium, that we actually had in one of the healthcare centers. And this analysis, I would say, don't use it to actually point to one culprit, because that's not why we should be using these kind of analysis, but it can really be used, and it's really important to actually evaluate and adjust local but also global safety measures. That's what is actually happening at the moment with the Delta variant, that many countries are actually changing their regulations to not have people accessing where there's a lot of prevalence of the Delta variant. So this is really based on the sequencing efforts done worldwide. Another thing we can do is look at the variants of concerns. As I said, the Delta variant here, this is an example of the UK variant, which became predominant earlier this year. If you look at the lower line, this was the normal mutation rate. So there was, over time, we saw that indeed we had the 2 mutations per month, but all of a sudden at the end of last year, there was a huge jump, and we saw that there was a new population here, which was then the UK variant. What happened there was an immunocompromised person, and the virus could all of a sudden mutate way faster than its normal mutation rate. This actually strain could escape from a hospital or a healthcare center, and this became actually a more evolutionary, better virus and could spread way easier. And that's how we can also check this with the data we have, by simply looking at the number of mutations at a specific time point. And then if you want to try this yourself and are really interested in it, I would definitely say don't immediately go to HPC for that if you don't have the data. But if you're really interested to have a look at these different variants and so on, I would recommend you to just have a look at the next train builds. It's very nicely shown. There's a lot of different features available. And this is actually the updated version of last week, where we can also see all the different variants present. We see that the beta variants and the alpha variant is still the dominant one. And then we have here actually the delta variant, which is the green one that is actually becoming the one of concern. This is a subsampling, so it's not all the data that is available at the moment. We also have views of this on Belgium, and these have been produced by Hibale from the KU Leuven, so they are also available on NextTrain. So definitely have a look there if you're interested in this. So that was about the COVID and how we use HPC. But as I said, I'm also working, and that's my major part on bacterial whole genome sequencing. So if we are talking about bacteria, we're talking about something bigger. If you see here, we have the viruses, and we have bacteria that are way bigger than the viruses, of course. And we're no longer talking about kilobases, but we're rather talking about megabase pairs here. So if we compare this, the E. coli, for example, it's 5 megabase pairs, then it's 166 times bigger genomes as compared to the coronavirus genome. So again, here, same story, bigger genome, more data. So if we first need to generate these genomes, it's already going to be a quite difficult task to finalize. Now, how is a standard genomics workflow using Nanopore data? We first need to base call the data. What does this mean? As you saw in one of my first slides, there was the current signal. So we actually got the power and the voltage changes. This is the raw data. This, of course, has to be converted to base pairs. So this is also called base calling. We call actually the nucleotides A, C, G, or T from the raw data. So based on how these currents look like, we will be able to tell which nucleotide is at a specific position in a single read that has been sequenced. Then we will demultiplex, because we don't sequence one sample at a time, but we will sequence multiple sequences at a time, so 12, 24, or even more. And then we will perform some quality filtering. We perform de novo genome constructions, which means we won't use any references that are available. So we use our data. And as I showed you the puzzle with the Wally, where we find Wally, we actually want to show this. We can construct this by sticking all the long reads together. And then there's a variety of downstream analyses, again, phylogenetics, but you can already imagine many genomes, and which are huge in size. It's going to be difficult to perform phylogenetics. Identification typing, but we are also interested in identifying and finding some virulence, but also the resistance markers, as I said, the antimicrobial resistance markers, genome annotation, and so many more. Now, long read sequencing data. This is also one of the advantages of it, is that for bacterial whole genomes, if you will look onto NCBI and you want to look into some specific bacterial genomes, you will definitely find them, but many of them will not be complete. Many of them will still be in the assembly phase, as they will only have an amount of separate pieces of the genome. So even though Illumina is highly accurate, it is not that easy in resolving the complete genomes. Now, what do I mean with that? If you look at the bottom picture here, you can see if we use the MinION that we will have a continuous contig, which means that we have one assembly for the genome. But if we look at MySeq, this is going to be all separate small pieces that are present. But some locations, it's not easily going to be resolved by the MySeq or Illumina data. The interesting thing as well with long-read sequencing, it's applicable to diverse bacterial species, as we can very easily perform de novo assemblies. And if you use Illumina, we will rather go to the reference base, where we use existing data to actually generate the genomes. With Nanopore, we don't need this. There's no PCR bias. We actually don't need to amplify our DNA. And if we use Illumina, we need to do this. And if we have a GC content, this means the amount of G's and C's within the genome. So I told you the code is ACTG, but depending on the amount of G's and C's, it's going to be more difficult for the enzyme to actually amplify. Now, in this case, we don't need this, and we can just isolate the DNA from our bacterium and we can immediately sequence that. So that's one of the advantages as well. It can resolve repetitive regions, which is also very difficult for an enzyme to accurately actually amplify, but also if we have very short reads and there's a long repeat region, the short reads won't actually be able to align properly because there will be too many repeats, and that will actually result in all these gaps that we have here. And another thing is it has advantages in metagenomics. Now you might wonder why there's a mycoplasma down here, but it's because it's one of my species of interest. And why is that? Because the genome, it's very low in GC content, which is already a peculiar one, and it has a highly repetitive genome as well, which makes actually these 2 points already it's difficult to sequence with Illumina. And there's very little complete genomes available for the Mycoplasma bovis, which is an important pathogen in cattle industry. So what is the diagnostics for Mycoplasma bovis at the moment? It's a very, very elaborate procedure. So it actually takes about 2 weeks before the veterinarian or the doctor, because we also have Mycoplasma pneumonia in humans, so you can extrapolate this, before we actually get a proper diagnostic result there. There are some quick identifications, which can be done with the qPCR. It's, again, amplification of a very small part of the genome. There's no sequencing, nothing. However, there's no antimicrobial susceptibility testing at all, which means we can't tell the veterinarian nor the medicine to actually tell him which antibiotic to use. And that's really the information that they need at the moment, because they want to know which antimicrobial drug to use. Because if you go to the doctor and they're going to treat you with another antibiotic, it can result in resistance of other bacteria in your body that you don't want them to become resistant. But secondly, it can also result actually in treatment of the wrong your mycoplasma in there becoming resistant to that antibiotic if it already has a kind of mechanism in there. So you will also not be happy, as the infection will take way longer than it should have been taken, because you want to be healed from the infection as soon as possible. So the quicker we can give a targeted therapy, the better. So what we actually did here is we actually shortened this workflow dramatically. We actually can do it now within 3 to 4 days, where we actually have immediately typing of the mycoplasma, but also immediately the genotype linked to the antimicrobial susceptibility testing. So we can actually see, based on the genome, if there is resistance towards one specific antibiotic or not. So again, here I will go into the importance of the HPC in these data analysis. Same story, bigger genomes, more data, more genomes, more data is, again, more computational power required. HPC has been very important for me, for us actually, for the base call training. As I told you, that's the conversion of the signal from the voltage output to really the nucleotides. Also, the base calling itself, it's a highly time-consuming process, and there's some GPU versions available which actually facilitate and speed up the processes. Genome constructions are important because at the end we want to have high accurate and complete genomes, but if you have hundreds of genomes to construct, HPC is very important. Philogenetic analysis again, and then a last one is Genomewide association studies, and I will come back to this later. So what we actually did at the beginning, we had the mycoplasma, but we saw with our data that the accuracy was not good, at least not good enough. So we wanted to look into some research here, and we checked some new base callers that were out there, and we found that that the Bonito base caller, it's just a program, a software. It can actually be trained to recognize the voltages based on the reference. So we can actually learn the program, we can teach the program to actually recognize the specific signals within the raw data to more accurately translate raw data into nucleotides. Interesting thing here is that this is also GPU based and that there's multi-GPU support. If we have to do this local, it takes a huge amount of time and it slows down the computer immediately. While if we can do this on HPC, it increased the speed of training. And what we actually obtained there, and that's also the paper that I state here, it's that we reached to genomes that actually resulted in consensus qualities of Illumina quality. So that's what we wanted to have at the end. Now, to really visualize this, what is really happening there and why the HPC is important, this is how these raw data signals are looking like. So it's a kind of voltage signal that comes out there. If we sequence this, there will be some mistakes. I've indicated these in the red here. If we look at the bottom in gray, we have the true reference. Now, if we want to train the model, we want to give the true reference back to this software and actually tell them, look, have a look, this signal corresponds actually to this motif. It's not going to look at single bases, but it's going to look at multiple nucleotides at the same time. We also call this k-mers. In this case, we have k-mers of 5 nucleotides. So it's going to tell the software, look, if we encounter in the data a voltage signal that looks like this, it's going to be this sequence. And the same will be done for all the different locations. So imagine this is not only for this small part, but we will have a lot of data in there. If we have a genome of 5 mega base pairs, this is going to be 1 million pieces that actually have to be aligned together with the true reference and converted. And in that way, we repeat the training that we really make the model smarter and smarter every time. And then we compare it to the real references that we have. Now, if we do that, we can actually see that the trained sequences after base calling with the trained model, it will actually be way more accurate. So the raw read data will be much better as compared to what we had with the default base calling models. Now, how does it end at the end? As I told you, Illumina is good for the accuracy and so on, but if you want to construct complete genomes, then this becomes rather difficult. If you look on the left and on the right, this is the genome of exactly the same Mycoplasma bovis strain. As you can see here in Illumina, due to the repeats, it is impossible to generate a complete genome. The software actually, it doesn't work. It can't find, it can't link the proper pieces together. So that's why you also see all these loops going back to each other here. The accuracy is good, but there's not really a complete genome here. If you look on the left side, we have the nanopore data. Originally, there was a quite big mistake, which means there were 1,000 mistakes in one genome. But with the trained data, we actually could get to 10 mistakes in one genome, which is the same as the Illumina data. And on top of that, we got actually the complete circular genome. And together with that, we also got the methylation, as you remember, the versatility of the data. Now, to end up of one of the interesting things we have here is a genome-wide association study. To do that, we need highly accurate but also complete genomes. I put a check mark here because that's what I just explained. We are able to get highly accurate and complete genomes with nanopore sequencing data only, but we don't want to look into one genome. We want to compare many different genomes of one species. In this case, for example, Mycoplasma bovis. Now, we want to identify specific genes or even point mutations. So point mutation is one single nucleotide within the 1 million base pair genomes that can be linked to antibiotic resistance or even virulence. Now, the more genomes we can enter, the higher the resolution will be, of course. Just a simple drawing here at the bottom. If we have 5 different strains of Mycoplasma bovis, and we know their phenotypes, in green they are resistant, this one is susceptible, we are able to identify specific sites within the genome. Either it is a single mutation, which is one nucleotide, or an entire gene, for example, which can be associated with the resistant phenotype. And indeed, if we, for example, look into 100 Belgian Mycoplasma bovis genomes, and we look at enrofloxacin, which is quite critical antimicrobial drug, which cannot be used easily in veterinary medicine as it is important for humans. We could identify specific mutations. You can see them here on the right side, which were linked and coming back in actually the resistant strains. The ones that are indicated at in orange at the bottom, these are actually the phenotypical, so meaning the really resistant strains. We could identify these having specific double mutations in 2 specific genes back. So that's valid and really important information that can be implemented in quicker diagnostic tests in the case for Mycoplasma bovis here. So that was my presentation. Thank you very much for your time, I would say. If there's any questions, I will be pleased to answer them now, or you can always contact me, or through HPC, you can also contact me. But this is my contact information. And once again, thank you to everybody from the HPC team for this opportunity. Thank you.
Thank you, Nick. Very interesting talk. Are there questions at this moment, Nick?
Maybe I have a question. I think you've done some work on the HIV virus as well, or at least some of your colleagues have. And could some of these techniques also be used there? And would that still be interesting at this point? Because that's already been going on for, I guess, about 30 years, maybe even longer. Are there still things you could learn there that's by applying these techniques that were applied to COVID, for example?
Yes, that's definitely possible. Like it's not my work, but the work from Laurens Lambrechts from the HIV Cure Center. The big problem with HIV is that within one single cell you can have a different virus, like a different variant of the HIV virus, and then accuracy becomes a very important thing, because if you're looking at one single cell within your human body, it's really important that the information of the virus that you get out, it's the most accurate at all. The big problem is that Illumina is also not always able to actually sequence these viruses properly. And the long read approaches actually can go and look at the entire virus in there. So we don't have to cut it into pieces. And I know that Laurens is also looking into this training of the models for the base calling to actually increase the accuracy. For example, these genome-wide association studies might also be interesting to link specific mutations or specific features of the virus to people that are more sick, people that have specific symptoms and so on. So I think it can definitely be done.
I don't see questions from other people. Okay, and yeah, okay. Celine has a. Go ahead, Celine.
Yeah, sorry, I missed part of the talk, so maybe you told it. But with the RNA-seq thing, you can also do direct RNA sequencing and looking into RNA modifications. Are you going to plan to do this, looking into RNA modifications of those viruses, for instance? It's like, for those that are not into biology, it's like the new kind of epigenetics level. So
Yes, that's a good question. Yeah, for me, I work at a virology lab, and we are definitely going to exploit that. But my project is more on the bacterial side. But I'm actually planning on doing some direct RNA sequencing this summer. So it's some really new technique, but I think it's really interesting to exploit in research. And I know that there's many people now also busy on the epigenomics of the SARS-CoV virus to really see if methylation patterns contribute to a more virulent strains. So I know that people are into that and busy with it. So it's like for HIV, it would also be interesting to go for it. But at the moment, you still need quite a lot of starting material. So that's usually the problem, that you don't get a lot of RNA from many samples. But it's definitely worth looking into it for viruses.
Maybe a question to wrap up if nobody else has a question. The use of GPUs, I think, for these kinds of training and analysis and to improve the accuracy of things is quite new, I think, in bioinformatics. Do you think this is picking up speed, and will there be more interest for using GPUs for these kind of things going forward?
That's a very good question. I know the Nanopore community is growing. Definitely know with the SARS corona sequencing, many people are like, it's an easy entry point for many labs for the Nanopore. And I'm quite sure that many more people will start to experiment on other levels with the Nanopore. And I'm definitely sure that there's going to be more people that want to experiment and actually really generate their own models and so on for accuracy. So it's also, if you look at the normal genome construction itself, there's many things that are available on GPU at the moment. There's polishing softwares available as well. And I know that NVIDIA is also working on a GPU-driven aligner, which is also quite interesting. So then people working with long read data, it's not only for Nanopore, of course, there's also PacBio and so on. These might really be interested in this in the GPU-driven resources.
Do you have an idea how much faster the Bonito runs are if you do them on GPU compared to CPU only?
Bonito, you don't have the CPU version. So Bonito really runs GPU, but the normal base caller, it was a difference of 40 times if you run it on CPU as compared to GPU. So if you really want to run the high accuracy models, because you can also have a quick and dirty model, then it goes very smooth. But of course, you want to have the highest accuracy, then it's a 40 times difference in time.
So GPUs actually enable you to do this. Without them, it would be impossible.
Yes, exactly.
Okay.
Cool. Thank you, Nick, for your contribution. Very interesting indeed. So I guess it's only remains for me to wrap up the meeting. Thank you all for your questions, your feedback, and your contributions. If you have any remaining problems, issues, questions, wonders, please do not hesitate to mail us at hpc@ugent.be. Thank you all, and see you in a couple. Bye-bye.
Cannot stop the recording.